program, and I will upload this to our YouTube page, the NPSO Emerald Chapter YouTube page, and I have a link to that in one of the slides here towards the end of my introduction. So I'll just move along with some of the introductions here. Let me get to the next slide. So we do have our next program is October 18th, and it's co-sponsored by the Cascade Ecological Society. We've been doing a co-sponsored talk for the past couple years. This year's talk is October 18th, so it's our normal program time, starting at 7. Plants that eat truffles for lunch. Dan Luoma will be discussing the fascinating plants that have gourmet adaptations, and you can get more information on our newsletter or on our Emerald Chapter webpage. But our next talk is October 18th. We have programs every month scheduled through March, the 3rd Monday of each month. So please be sure to join us if you can, and we will continue to record them and post them on the YouTube channel. Also, we do have a field trip coming up October 10th, Sunday. Whitey Luck will lead us on a tour of trees on the University of Oregon campus. I'm not exactly sure if that trip has been filled completely. There is a limit, but if you want more information, please go to our chapter website, and there's other details, and if there's a space open, you can join. But we are still conducting field trips, and we hope to have some more throughout the winter and spring as the year progresses. Just some other Emerald Chapter happenings. We're always looking for, if you're interested in getting more involved in a leadership position with our Emerald Chapter, you can join us for a board meeting to just see what we're working on, or to plug in if you want to, or where you can. If you're interested in plugging in at the more leadership or board level, I should introduce myself also. I'm Stephen Yeager. I'm the current Emerald Chapter president, and you can reach me at the email address there. It's em_president@mpso-oregon.org. And if you want to be more involved in the workings of our chapter, board meetings, or participate, we might have some in-person events coming up, as far as maybe the Mushroom Festival. But also, we are looking for a program chair and a publicity chair. We do have currently have those positions filled, but if we're looking to mentor some new members to participate in those areas. Please mute. Can I hear speaker? And okay, and then lastly, the Mushroom Festival will be taking place, and it will be a smaller live event, and it's Sunday, the 31st. Tickets will be by pre-sale only, and I put just the list of what they had on their website. But the Mushroom Festival show will be happening. I think they crossed out the word festival and calling it more just the mushroom show, because they are expecting to have a smaller event. But this will be an in-person event. I think one thing to note is that tickets will be by pre-sale only, which is different than past years. So, okay. And then lastly, before we introduce our speakers, just kind of some housekeeping. Please, we're going to take questions at the end of the program. So if you want to post questions in chat, I'll be monitoring the chat, and then I'll present the questions at the end to our speakers. And then, like I said, we will also open it up at the end if you want to open up your video, your audio, and present a question live. Please keep your sound and your mute. Please keep it on mute and videos off. It just helps keep it less distracting and lessen some of that background support, background noise, I mean. And then also, I just want to thank you all for supporting the Native Plant Society of Oregon by even coming to these programs. And if you're not a member, I recommend becoming a member. We'd love to have you. And you can reach out to me at the Emerald Chapter President email, or you can also go to our state webpage to look about becoming a member and look at the work that the Native Plant Society of Oregon is currently working on. You can see there our Emerald chapter website link is there, and I also posted our YouTube channel link where I'll be uploading the recording tonight. And we also have a few other recordings on there from last year at this point. Okay, and with that, I'm going to pass it off to Gail to introduce tonight's speakers. And then Gail, you can share your screen whenever you're ready to do that.
Okay, there's two Gails involved here.
Yep.
So I'm also welcome to everyone who's joining us tonight. I'm so glad you're all here. And my name is Gail Baker. I'm the program chair of the Emerald chapter, and I'm the vice president of the state organization of the Native Plant Society of Oregon. So I'm really happy that you're all here. I'm happy to be here and looking forward to the presentation tonight. We have lined up some really nice presenters, some exciting presenters through April of this year. And so hopefully you can join us for all of them. So I'd like to introduce Gail and Jen tonight, both from Oregon State University. And they're going to talk about native plants for pollinators, and specifically research that has been done in the Oregon State University Garden Ecology Lab. And what's interesting to me is that they're going to talk about not only the native plants, but also cultivars that have been developed from the native plants, and the relationship between not only the pollinators and native plants, but pollinators and these, you can correct me, Gail, nativars. Is that correct? Okay, so that's really interesting. This research is fascinating. And so Gail is the professor of horticulture and the statewide coordinator of Oregon's Extension Master Gardener Program. And with her lab, the Garden Ecology Lab that I mentioned earlier, she studies plants, insects, animals, people, decisions, and management practices that either improve or degrade a garden's ability to promote environmental and human health. So that whole infusion and integration of humans and natives and the landscapes and gardening we care about is very interesting to me, and I like that holistic picture. Jen is a graduate student in the program, pursuing her PhD in horticulture and entomology at Oregon State. And Jen hails from Vermont. So welcome to Oregon, Jen. And she's passionate about pollinators. She had her first exposure to bee research while she was working in pollinator studies in Vermont, but she's also studied in Ecuador, North Dakota, and now in Oregon. So I'm ready to hear this program, as we all are, and thank you very much for coming. And thank you all, the audience, for coming. Thank you so much.
Thank you, Gail, for the introduction. Stephen, for inviting us. I believe that August originally extended the invitation, so I would like to thank him as well for inviting us to talk a little bit about our research. And I just want to say to Bert, whoever you are, best avatar or Zoom photo ever. I love the cats and planets. So I'll be starting us off, and then I'm going to hand over to Jen to finish up the second half of this presentation. We do look forward to your questions at the end of the talk. In case you want to follow up with either of us at the end of this talk or after this talk sometime later, here's our contact information. We'll show this again at the end of the talk as well, in case you see something you wanted to follow up on, ask a more in-depth question, just get in touch in some way, shape, or form. So even though we're going to be talking about some of the research that we're doing in the OSU Garden Ecology Lab, as somebody who works with gardeners all across Oregon as a primary part of my position, I know that native plant gardening is something that is really at the forefront of many gardeners' minds. They're very interested in learning more about native plants that they can use in their garden, in large part because of purported benefits of native plants to pollinators. So this presentation is going to be divided into 3 parts. First, I want to give a little bit of a primer as to why native status should matter to insects in the first place and why they might matter in somewhat different ways to herbivorous insects versus pollinating insects. Second, I want to give a little bit of an overview of some of the research that we've done on gardeners and their preferences for different native plants that are sold in home and garden stores. And then Jen is going to take us home by talking about her really innovative research on native cultivars, native plants, and pollinators. And I always like to put the punchline at the start of the talk, just in case for whatever reason, you want to tune in for the beginning, but there's a really great show that comes on at 8 p.m. This is a summary of 3 years worth of research that we've been doing on native plants and bee associations. This particular research I'm going to talk about a little bit more later in the talk, but if you just wanted recommendations for good native plants that look beautiful, are well suited to garden landscapes, and that support bees specifically, here are the top 10 plants that we are recommending. The name of the plant, both the common name and the Latin name, are listed in each box. And the 2 circles that are superimposed on top of every plant represent 2 different pieces of information that we gathered from those 3 years of research. The outer ring over every flower represents the relative abundance of bees on a particular flowering plant, and the inner circle represents the relative abundance of species, so the number of different species that we found on different native plants that we tested. These top 10 are a subset of 27 total plants that we tested for associations with bees. And you can see that some, like Lobelia and Dugaster, have both high bee abundance and high bee species richness. Others, such as yarrow, have relatively lower bee abundance, but have relatively high bee species richness. So since this is the Native Plant Society of Oregon Emerald chapter, I'm assuming that you are lovers of native plants, and you may very well have some of these plants in your garden already. If you don't, and you're looking for more natives to add to your garden, here are the top 10 that we recommend. And the subsection headings that I'm using between each of the 3 sections that we're going through today are my 4 personal favorites, both because of the incredible bee biodiversity that we found on each of these 4 native plant species, as well as I just find them to be incredibly lovely, and I just don't know why anyone wouldn't want them in their garden. A little bit of a background on native plants and insects and why we might expect that native plants would be better for insect pollinators than non-native plants. A lot of the idea about why native plants might be better for pollinating insects such as bees relative to non-native plants comes from what we know about associations between native plants and insect herbivores. About 50% of all insect species are herbivores, and of those herbivorous insects, we know that about 70% of them are specialists that are only capable of feeding on one plant family, plants within one plant family, or in some cases, even more narrow, only capable of feeding on plants in one genus. So of course, one of the classic examples of this relationship is the monarch butterfly caterpillar, which is only capable of feeding and developing on plants within the genus Asclepias. Many gardeners will also know cabbage white butterflies, which can be pests of kale, cabbage, other brassicas. This particular insect, even though non-native, has a specialist relationship with brassicaceous plants. Cabbage white butterflies are only capable of feeding and successfully developing on plants within the cabbage and collard family. And this relationship between specialist insect herbivores and native plants was really kind of captured and communicated in Doug Tallamy's amazing book, Bringing Nature Home, where he makes a really strong and compelling argument for the need to include native plants in our suburban landscapes if we want to sustain wildlife in our gardens. A lot of Tallamy's research came from separate studies that he was doing. On the left-hand side is the capture of a field study that him and then graduate student Corinne Burkhart did in Pennsylvania, where they looked at the abundance of caterpillars in Pennsylvania gardens that were landscaped predominantly with native plants or predominantly with exotic plants. And they found that gardens that were landscaped predominantly with native plants had significantly more caterpillars than those which were landscaped in more traditional fashion using non-native ornamental plants. And what was really groundbreaking for me about this particular study is that they showed that that strong relationship between caterpillars and native plants actually cascaded up one trophic level and resulted in a higher abundance and a higher species richness of birds in gardens that were landscaped predominantly with native plants. And then Doug Tallamy and another colleague combed the literature to try to find associations between caterpillars and plants, specifically ornamental plants. And they found when they were looking at the abundance of caterpillars on non-native, exotic, introduced ornamental plants, that overall abundance was relatively low. But when they started looking at native plants, they saw that the abundance of caterpillars increased by 14 times if you were looking at woody ornamental plants, or 3 times if we're looking at all plants combined. And the reason that native plants are thought to promote such high biodiversity of insect herbivores in particular is because there's this strong evolutionary relationship between plants and their herbivores, which promote specialist feeding habits between insect herbivores such as this cabbage white butterfly caterpillar and a narrow range of host plants on which they can feed. Plant defenses are going to deter most herbivores, but specialist herbivores have the ability to crack that chemical code of plant defenses that are being put out there to dissuade most feeding. And that encourages strong specialization and ties between native herbivores and native plants. However, the story might be a little bit different for pollinating insects. Whereas plants want to keep insect herbivores from feeding on the vegetative tissue, plants actually advertise and try to draw in pollinating insects so that they can help the plant to reproduce. And for that reason, there has been some disagreement over time as to whether or not native plants necessarily would promote strong specialization by pollinating insects. And in fact, before I came to Oregon State, I was an assistant professor at Fordham University in New York City. We did a series of manipulative experiments adding native plants to community gardens in the South Bronx and in East Harlem. We wanted to see whether or not adding in these butterfly conservation areas composed of about 70 to 100 native plant starts would result in any change in the insect community in these gardens. And it did not. Over 3 years, we started to see that there might be a little bit of a difference in butterfly abundance, but it wasn't significant. And in fact, 88% of all of the butterfly observations that we saw in these gardens were butterflies that were nectaring on non-native plants. We also found that leafcutter bees were found to visit non-native flowers more than native flowers, and we found no difference in total bee abundance, bumblebee abundance, or honeybee abundance on native versus introduced flowers. A couple of caveats about this particular study. First of all, it was conducted in a hyper-urban area of New York City. Second of all, a lot of the insects that we found in this garden and that we observed for this particular study were non-native insects themselves. The majority of butterflies in the study were non-native cabbage white butterflies. The major leafcutter bees that we saw were non-native rotund leafcutter bees. And of course, honeybees are a non-native bee to North America. Still, because of the generalist feeding habits, or the supposed generalist feeding habits of pollinating insects, I wasn't really sure whether or not native plants would be preferred by native bees over non-native flowering plants. But when my first PhD student at Oregon State decided that they wanted to study native plants and pollinators, I said, go for it. I'm a big believer that graduate students should study something that they're passionate about. It'll make graduate school that much more fun and easier to get over some of those stumbling blocks that are necessarily a part of graduate school. So this is the work of Aaron Anderson, who you see on the right-hand slide in the foreground. Lucas Kostner was an undergraduate field assistant who helped him for 3 years over the course of this study. What Aaron did is he planted out 27 different plants at the North Willamette Research and Extension Center in Aurora, Oregon. These 27 plants were planted out in 1 meter by 1 meter square blocks. Each individual plot of plant was separated from its nearest neighbor by at least 6 meters to give some level of independence. And in addition to having the 27 plants that Aaron tested, he also had 1 meter square plots of just turf, which served as a level of control. So in total, he studied 140 plots over 3 years, monitoring bee visits as well as overall insect abundance and visitation as well. To look at bee abundance, Aaron and Lucas, every time a plot was approaching peak bloom, would sit and observe whatever insects came and visited a flower for 5 minutes for each plot. They did this on days when conditions were good for pollinator counts, which basically would not be a day like you see on the left-hand side of this slide. In general, if they could see their shadow, if it wasn't too windy, then that was a good day for observing bees. In order to get bees to the taxonomic level of species, Aaron also had to collect them to take them back into the lab. He used this modified insect vacuum to plop in 4 separate places on each flowering plant plot so that he could census all of the insects that he found on each flowering plant plot when he took them back into the lab for identification. Just to note that overall, throughout the course of his study, Aaron found 56 different species of bee on his study plot. But I also want to give a shout out for all the work he did to identify all of the other insects that were not bees. He collected over 40,000 individual insects, and he identified all of them at least a family, and most of them at least a genus. I'm really excited for that data, which should be coming out shortly. And this is just an overview of one of the bee abundance data plots that Aaron generated throughout the course of his study. This is a summary plot of the 2019 field data. On the y-axis, we have mean bee abundance during the 5-minute counts. On the x-axis, we have the individual plant species on which Aaron was monitoring bee abundance. When the plants have a red asterisk above them, that notes when they were a non-native plant. Whoops, sorry, these got shifted a little bit, so not all of the asterisks were over the right place. This asterisk right here should be over Nepeta. So this one goes over Nepeta, and then this one right here should go down over Salvia. Sorry. So what you'll notice is that the highest bee abundance were on 2 non-native plants, oregano and lavender. And I'll note that we included 4 species of non-native comparison plants because we really wanted an easy way to communicate with gardeners who might observe just casually that they have a lot of bees on a plant such as lavender. We really wanted to be able to say to them, well, you think lavender has a lot of bees, just look at this other plant. So these non-native plants were included as comparison species. So even though we see that both oregano and lavender have significantly more bee visits than the other plants in the study, when I tell you that this yellow-orange part of the bar stands for the amount of honeybees that visited each plot, it tells a very different story. Of course, honeybees are a non-native bee to our region. If we're just focused on the native wild bees that were visiting the different plots, a very different story emerges. Phacelia, California poppy, goldenrod, Doug aster become the plants which have a higher abundance of wild bee visits. So one of the other studies that we've done in our lab, and I'm not going to talk about it tonight, but I did go around and sample bees from Portland area gardens for 3 years. And something that I noticed that gardeners would always say is like, you know, go to my lavender, go to my oregano, they're just hopping with bees. And that is true. Gardeners will see a lot of bees on their oregano, on their lavender, on their catmint. However, if you look a little bit more closely, you're probably going to notice that the majority of these bees are non-native honeybees. And does that really matter? Well, some folks may suggest that it could. Honeybees are such efficient foragers of nectar and pollen that they may have negative impacts on native bee populations. Specifically because honeybees are such efficient foragers, they might deplete nectar and pollen from plants, which might reduce native bee reproduction. This is because native bee females are going to have to use more resources and more energy to find other resources further from their nest. If female bees have fewer resources, that could lead to a skewed sex ratio towards more males and against more females. If female solitary native bees have fewer resources to provision their offspring, that's going to lead to smaller offspring, which have a higher chance of not surviving. And the longer time a female bee has to spend away from her nest, traveling to look for nectar and pollen, that actually increases the vulnerability of her brood to parasites. So once again, if you are interested in planting native plants to support native bees, here are the top 10 that we found to have the highest bee abundance, as well as the highest bee biodiversity. And specifically, these are the plants which were not super attractive to honeybees, but were very attractive to Oregon's wild, mostly solitary bees. If I were to put lavender on this particular graphic, what I would see is that lavender would have an abundance ring, which is as full as Douglas aster's abundance ring. Bees are very, very abundant on lavender, but most of those bees are the same bee, the non-native honeybee. So if we take a look at species richness on our top 10 native plants for pollinators, and we compare that to species richness, or the number of different bee species on lavender, we see a very different story. On lavender, we found a total of 8 bee species, and each of these little circles represents a unique species of bee. On Verily Phacelia, we have twice that number, 16 bee species. Glogilia, 28 bee species. Douglas aster, 74 bee species. So many of these little circles representing a different bee species that we can't even see the aster anymore. So with a pollinator super attractor such as lavender, you are going to get a lot of bees, but they're not going to be the richness and diversity of bees that we see with our native plant species. I want to spend a little bit of time talking about native plants and gardeners before handing it off to Jen to talk about her really innovative research. And once again, these 4 flowers are my personal 4 favorite native plants for bees in our area. If you've ever tried to find and locate native plants from local nurseries, most gardeners have the experience of having a limited selection of native plants for sale at local nurseries. In general, this limited availability of native plants in nurseries leads to an overall lack of familiarity and lack of knowledge by gardeners for native plants, and tends to promote a lack of preference for native plants by most gardeners. You can't prefer what you don't even know about. Because there tends to be a relatively limited audience for native plant purchases, a lot of nurseries don't invest the time and energy to propagate native plants for sale. Because of the limited market for native plant sales, nurseries will also say it's hard to sell native plants at the point of purchase, because when you put a demure native plant next to a showy or ornamental, many consumers' eyes are automatically going to be drawn to that showy or ornamental. It's just harder to get native plants to sell off of the shelf, particularly for perennials, which don't look their best until several years after establishment. We're trying in our lab group to work with gardeners and help them see that the beauty of a plant really isn't just skin deep, but there's something that we're calling the ecological beauty of a plant. It's not just how beautiful the plant is itself, but really all of the other organisms that a plant can bring into your garden should be part of the beauty of that specific plant. I had this slide up here because I saw this meme on my social media and I saw this quote and I was like, oh my God, I love that quote. And then I realized like, oh, wow, I guess I said that quote at some point in time. But it is something that we're trying to promote. Ecological beauty is as important, if not more important, than the aesthetic beauty of a plant. We did some consumer testing and asked folks to give us their impressions of the native plants that Erin studied. Here we have Verily Phacelia, which ranked dead last of all of the plants that we asked gardeners to chime in on what they thought about a particular plant. Most gardeners thought that this plant it looked weedy, it wasn't attractive. They were convinced that they would not have a place for this particular plant in their garden. But when we showed them the bee species which were significantly associated with Verily Phacelia, longhorn bees, Bombus mixtus, this Panurginus bee, other species of native bumblebee, then that significantly changed their impression of this plant. And it still did not make Verily Phacelia the top runner for most gardeners, but it did convince a significant majority of gardeners to at least give this plant a try. I like to say that Phacelia really just needs its own PR campaign, so I wanted to spend a little bit of time giving a little bit of love and PR for 2 other plants which ranked very low on our survey of gardeners and what they thought of native plants for garden settings. Pearly Everlasting also ranked very, very low in our survey. I don't understand why. I think it's a beautiful plant, but many gardeners thought that this one just looked too weedy and that it shouldn't have a place in their garden. We found 21 different species of bees on Pearly Everlasting, including these Andrena bees, which were significantly associated with Pearly Everlasting. And then Goldenrod, which a lot of gardeners don't care for, and I don't understand why. I think it's so beautiful with the fall profusion of bloom. 14 different species of bee on Goldenrod, including a significant association with Melissodes microstictus, which looking at biodiversity databases is a relatively rare bee. So I try to tell gardeners, Goldenrod, I think it's beautiful. Maybe you don't think it's the prettiest girl at the pageant, but if you plant it, you're going to be more likely to attract in a rare and special friend in the form of this longhorn bee. And I think with that, I'm going to hand it over to Jen.
you. Going to just get this set up here. All righty. So my name is Jen, and I am a grad student working with Gail, and I'm very excited to be here because I have lived in Eugene for all of my Oregon life, which is subject of conflict typically when people find out that I go to OSU, but I promise my partner went to UO, and I love Eugene more than Corvallis. So all that aside, I'm going to start this last section of our talk with a brief overview of some traits that are important to consider for pollinators when gardening for them. And then the focus really is going to be on what we know about cultivars and our current research. Alrighty. So many plant guides recommend selecting plants based on color, and it's generally accepted that bees prefer plants that are white, blue, yellow, ultraviolet, and then butterflies prefer reds, magentas, purples, and other bright colors. And although color can be a helpful tool in guiding selections, there are other tools that actually might be more important when planning for pollinators. So of course, this talk is about native plants, so we're not going to talk about native status too much here, at least. But in general, when creating native pollinator habitat, perhaps the most important consideration is selecting blooms that bloom across the season. So things that bloom from early spring to late fall. And doing a little research to determine the nutrition that your selected plants provide to pollinators is also something to consider. So for bees, we want to make sure that they have access to pollen and nectar throughout the entire season. If you were to use this planting scheme that I put along the bottom of the page, you might actually find out that California poppies don't produce nectar, they only produce pollen. So if you have a time of year where poppies are the only thing blooming in your garden, you might actually want to complement them by adding a plant that you know to be a good nectar source, which is where some of the non-natives can come in sometimes. But if you're more interested in attracting butterflies, you should also think about including both nectar and host plants in your garden. So a quick Google typically can tell you if your butterflies of interest are generalist egg layers, or if they have specific or preferred larval hosts, such as monarchs and milkweed, which Gail mentioned previously, or pearly everlasting for American lady butterflies. So that's all I'm going to talk about for just general planting for pollinators. And now we're going to move on to natives and cultivars. So if you've ever heard the term nativar, it's just a combination of native and cultivar. And just like cultivars, you can identify nativars because they have that little single apostrophe around their like fancy industry name. So here we have 3 examples of native cultivars of Clarkia. All 3 of them are from Silver Falls Seed Co. and they're very beautiful. And I just wanted to provide like a visual example of what nativars can look like, although you'll see plenty in the rest of this presentation as well. And you can use native cultivar and nativar kind of interchangeably. I probably will for the rest of this talk, just so you know. And like other cultivars, oh, sorry, I already said that. Getting ahead of myself here. But yeah, so you can see these single quotation marks followed by the scientific name, or sometimes in nurseries, you'll just see the common name of the plant. So I want to talk a little bit about why nativars exist in the first place. So of all of the plants that are sold in the US horticultural markets, only 13.4% of them are reported to be native, but of that 13.4%, much fewer of those actual species may be true natives, since many of the genotypes that we actually find in the markets have been bred or selected for ornamental qualities. And again, like Gail mentioned before, some natives just don't respond well to nursery plant production. So they can look really sad and droopy in retail containers and fail to meet customer expectations. You know, if you're going in and you want a beautiful aster, you're probably going to choose the one that has some flowers on it versus one that's kind of brown and falling over. So the development of nativars is one potential solution to this barrier of native plant production, as nativars can be easier to propagate in large numbers and are often perceived as being more attractive in containers and in landscapes. So I'm going to talk about some traits that we can see altered in the plant breeding process. And I also want to start just talking about natural variation. So we know, especially if you've ever tried to key out some of our more diverse plants, that we see a lot of variation in plant morphology just out in the wild. So for example, Clarkia sometimes doesn't have those little red maculations at the center of the plant. Sometimes it's just a total pink petal. And a study of Clarkia with and without these spots actually found that the presence or absence of spots does not impact the insect visitors that go to that plant. And another example of natural variation is hydrangeas. I'm sure you've all seen or tried to change. The soil pH can change hydrangeas from anywhere from white to blue to purple. And so nativars may allow nurseries to capitalize on positive traits of native plants that gardeners may find attractive, such as isolating rare color morphs that are naturally occurring, such as white camas or even white bee balm occurs occasionally. And these ornamental characteristics can be achieved through different breeding techniques, some of them including mutations, actual interspecific breeding, changing or multiplying genes, and then also in selecting for function loss in particular genes. And so color is actually genetically very simple, but dramatic changes in color can impact plant and pollinator interactions. So all it takes is a single gene change for this white petunia to start exhibiting this bright magenta color. And that significantly alters pollinator preference. So prior to the gene change, the primary visitor to this petunia is a hummingbird moth. You can see here it very drastically outcompetes the bumblebee here in visitation and feeding to the white petunia. But after that gene shift, the bumblebee becomes the primary visitor of the flower, and the hummingbird moth almost totally stops visiting it. So one gene change and this plant pollinator interaction is significantly changed and interrupted. So I also want to just highlight a study that has really been central to the foundation of our own native and nativar research. Annie White did a study at the University of Vermont using Vermont native plants and nativars as part of her PhD project at UVM. So she had 19 different native and nativar pairs and tracked pollinator preference between the pairs. And she also did included a few more cultivars of echinacea, which are here on the bottom. And so I want to walk through her findings. And we're just going to consider her data when only bee visitors are considered. And first, we're going to exclude 3 plant pairs here where the data was not published. So we're going to start with, I have these little color-coded bees on the left side, and green corresponds to there was a preference for native in that pair. If there's yellow, there was no clear preference between the native and the cultivar, and then this pink is going to highlight couples where there was a preference for the nativar. So here's the green. And we can see here that 6 of her plant pairs showed pollinators significantly preferred the native plant over the nativar. There was no significant preference for 5 plants when bee visitors are considered. And there was only 1 species pair where a cultivar was actually more attractive to pollinators than the native. So only other studies looking at natives and nativars are still in progress, but many of the ones that have wrapped up or have published partial data have had really mixed results, which just highlights the need for further evaluations, which is where we come in, at least for the Pacific Northwest. So we are actually in the 2nd year of our own study looking at pollinator preference for Willamette Valley native plants and cultivars. We have 2 main research questions. The first being, are native plant species more attractive to pollinators than native cultivars? And then our second question is, if there is a difference in visitation between the natives and the cultivars, can we try to find out what traits might be responsible for those changes? And so we have 3 different kind of overarching hypotheses about what we expect to see from our plant groupings. So I'll start with positive response, and this would be a scenario where the cultivar was more attractive than the native. And so we think we might see this in situations where maybe the plant has a bigger flower size, which kind of increases its ability to signal for pollinators that it's there, or maybe even situations where the cultivar has a lot more blooms on a single plant than the native. We also expect there might be cases where there is no difference in pollinator attraction. And we think that might happen in cases where the plants are very, very visually similar. So they almost have no clear difference to the pollinators, like between the flowers. And we also think that there are cases where we will see a negative response. So where the cultivars are much less attractive than the natives. And we expect to see these, especially in cases where maybe the flower color goes outside of the typical pollinator syndromes. So in this example, we have baby blue eyes, which is Nemophila menzesii. And one of our cultivars is this kind of purpley black color, which you typically don't put in your garden if you're trying to attract bees. So we think we could see some, definitely a few negative responses. Our experimental garden is set up fairly similarly to Erin's study, except all of our plants are right next to each other, but it is located at Oak Creek Center for Urban Hort, which is on OSU's campus and open to the public if you ever want to come take a look. I selected 8 plants from Erin's study that had cultivars available in the retail market. And we also picked plants knowing that they existed on a spectrum of different levels of attractiveness to pollinators, so low, moderate, or highly attractive. And our goal there was to see if maybe a plant that wasn't very attractive in Erin's study, one of those cultivars has a new trait that might make it actually very attractive to pollinators compared to the native. And so I'll just name these pretty quickly with their common names, since I just put the scientific ones here. So across the top, we have our perennials: yarrow, western red columbine, great camas, Douglas aster. And then our annuals: farewell to spring or Godacia, California poppy, and baby blue eyes. And so our cultivar selection process was really guided first and foremost by what cultivars we could find for our natives. And then if we had plants where there was a wide variety of options available, we tried to pick out different traits. So for example, there's like thousands of cultivars of yarrow. So we picked 2 that have very different colors than the native, and then one that has a different foliage color. And then on the other end of the spectrum, for our Western red columbine, there are almost no cultivars available. This Zerotones cultivar just has like maybe a little pinch of our native in its actual genetics. It's mostly been bred from a European variety. So I just wanted to also quickly highlight some other traits that you might not be able to see. It's really easy to see the color differences here, but we also have plants, again, with different foliage colors. So our white camas actually has variegated foliage. Then we also have some plants where the bloom density is very different. So here's our native California poppy compared to purple gleam. And then we also have some plants where the actual height of them is drastically different. So this is a photo of me, and I tried to make sure they were to scale, all of the 3 little gens, with the native Douglas aster here on the left and our 2 cultivars on the middle and the right. And so other differences that we see between our natives and cultivars are actual bloom size and differences in bloom period and duration. And I realized we are getting pretty close to 8 o'clock, so I'm going to try to pick it up a little bit here as we breeze through methodology. So while our plants are in bloom, we work to measure 3 main variables to quantify the differences between the natives and the cultivars. The first is the actual insects that come and visit our plants. So like Erin's study, we do physical collections of specimens so we can ID them down to species or higher taxonomic levels in the lab. And we also do visual observations where we sit and watch the insect visitors to our plants during a 5-minute period. We also are measuring aspects of floral display, such as the physical size of the plant, so the corolla width and depth, as well as the total number of blooms that each of the plants produce. And then we're also measuring the duration that all of our plants are in bloom. And then we are also looking at, we're using a specialized form of photography to try to mimic bee vision to see if there are any differences in the plant groups that are only visible to bees. And then finally, we're also going to be measuring aspects of floral rewards. So we'll be analyzing pollen from a subset of our plants to see if we see any differences in pollen nutrition between the natives and the cultivars. And then we're also going to be looking at the sugar content of the plant nectar for a subset of our plants.
Hey, Jen, I'm just going to say, don't feel rushed. We have plenty of time.
Okay, I kind of just as this was an hour, but I will slow down my talking voice then. I did want to briefly highlight the work that an undergraduate in our lab is doing as part of our project. So Sabea Breslin is handling the multispectral photography, and she uses a combination of filters to achieve bee vision. So as you might imagine, we don't even have the same kind of receptors that insects do to communicate with and see plants. So bees see largely ultraviolet, blue, and green light, which we are achieving through using these fun combinations of filters. And so again, the goal of these photos is to try to capture any ultraviolet messages on our plants that might only be visible to bees, and to see if those might be altered in the plant breeding process. So I just briefly have one example here. This is just the native baby blue eyes photographed 3 different ways. And so the first is visual, which is how we perceive the plants. The second is just ultraviolet. And then the third is this bee vision, which includes the ultraviolet as well as the blue and green. And what we see here is what we call a bullseye. So you can see nectar guides, sometimes they're visual, like the little spots on irises and mint flowers. But in general, it's kind of like a very contrasting color on the plant that helps guide insects and other pollinators to plant resources. So here we are seeing a dark contrasting color in the center of the flower, and the bee vision one is a little harder to see, but it comes through as this cyan color. And what we actually have found is that plant breeding can change these signals. So one of our cultivars of baby blue eyes is totally white, and we still see this little cyan circle in the middle of the flower, versus that black one, which I showed you on the hypotheses slides. It actually has kind of like a double fluorescence. So it has a little circle in the middle, but then the tips of the flowers, of the petals where they are white, also fluoresce under this B vision. All right, so now moving into preliminary findings, I just wanted to give a quick overview of our actual sampling effort between year 1 and year 2, because both years we've kind of had some weird weather stuff to deal with, like the fires last year, and then the incredibly hot June that we had this year. But luckily we were actually able to sample a similar number of dates between both years. And this is the number of days where we've gone into the field and sat and done observations or collected pollinators. And then we're still working through our collected specimen from this year, but last year we caught over 2,000 pollinator specimen off of our plants, and we're expecting it'll be pretty similar this year as well. And then when we've sat down and done those visual observations, we have made over 6,000 records of pollinators interacting with our plants for each of the 2 years of our study, which is pretty exciting to be able to say. And so I'm going to walk through our very preliminary findings with you, similar to the way that I did with Annie White's study. But I do want to really underline the fact that this is preliminary, and we're still actually sampling this year because the rain has really excited the Douglas aster into flowering for a few more months. So this data that I'm going to show you is based only off of the observation data, so none of the physical specimen that we've collected. And it's just been calculated from the average number of native bees that visited the plants during our observations over the last 2 years. So immediately we're ignoring flies, butterflies, honeybees, and other non-native bees with these metrics. So for 2021, we had 3 plants where there was a clear preference for the native, and that's in the columbine, the clarkia, and the California poppy. We had 2 plants where we didn't have a very clear preference, and that is sometimes due to just a lack of data, and other times it may be due to just all of them having the exact same average. And those are the yarrow and the Douglas aster. And then we did have 2 plants where a cultivar was found to be more attractive. So in the case of camas, it was this white cultivar, which was more attractive, which I think is likely because it blooms for a lot longer than either of the 2, the native and the other cultivar. And then unfortunately, the Nemophila is marketed as like a full sun plant. And I don't know if any of you have ever tried to grow it, but I don't believe that at all. It has had an impossible time growing in our full sun study plot. So we've just had a really hard time getting data off of it. So I will say take the Nemophila data with a grain of salt. We might end up having to exclude it just because it performed so poorly. But based on the data we were able to collect last year, the Snow White was the winner of the group. And we expect there to be some variation in year to year in this data. But one thing that Gail said a few times in preparing for this study is, in terms of perennials, the first year they sleep, the second year they creep, and then the third year they leap. So we expect that as our perennials mature, we might see some changes in pollinator attractiveness. So maybe by year 3, the native yarrow will just have way more flowers than the cultivars, and maybe it'll win then, but we do expect there to be some variation in the perennials. Whereas with the annuals, they should have similar responses every year, except for changes that might be due to natural fluctuations in the actual pollinator community. And so with that, I will show you this year's tentative data. And you can see that a lot of things are the same, but there are a couple changes. So this year, it seems like the native yarrow beat the cultivars. And again, the Nemophila, no preference, but it's not doing well in our study. But otherwise, everything is the same. I did want to note, I've run these analyses with the different combinations of pollinators that we looked at in our study. And basically, if we include all the pollinators, so if we add back butterflies, flies, honeybees, and it really is honeybees that makes up the majority of excluded observations here. The only change, at least in this year's data, would be for the Douglas aster. So if we add all of those pollinators back, we find that the Savi snow actually attracted a lot more pollinators than the native or the Savi sky. Okay, so to start to wrap things up, I know it can be pretty difficult to interpret these findings and kind of how to understand how you can apply them to your own gardening decisions. So I wanted to end with some guidance. Obviously, I think y'all might be a little biased because this is a native plant group. But in general, I would recommend consider what your gardening goals are. So if you are a die-hard native plants only person, nativars obviously might not have a place in your garden. And that might also be true if you're trying to create really intense pollinator habitat and you're trying to conserve really sensitive species. Nativars probably would not be a great choice for you. But if you really love Oregon natives and gardening for aesthetics, you might be super thrilled to find out that some of your favorites come in other colors. And you should also consider what plants you have access to. So native plants can actually be really difficult to find, and maybe you don't have a native plant nursery in your area. Not everybody has the time or resources to go out of their way to, for example, drive to Portland just to buy some plants for their garden. And instead, you might end up only having like Lowe's or Home Depot that you can shop at for plants. So you might only have cultivars available to you if you even have natives in your area at all. And I just wanted to highlight that just because some of our nativars were less attractive than their natives, that doesn't mean that they didn't receive any visitation. In some cases, these plants may still be a great pollen or nectar source, or in the case of camas, it actually, you could use a cultivar to extend your bloom period. So the native camas blooms first and it blooms for a really short period of time. And the white camas actually blooms like a week and a half later. So if you really love camas, you could extend how long you have camas growing in your yard by choosing a cultivar that has a different bloom time and different bloom duration. I would say right now, probably the biggest concern of gardening with native cultivars is the potential for them to hybridize or interbreed with their wild counterparts and potentially become the dominant genotype in the wild. And this could especially be problematic for the conservation of sensitive pollinators or ecosystems where hybrids cannot support specialist insects, such as the hybridization that we see with Kincaid's lupin and the Fender's blue butterfly. Personally, I believe that cultivars may have their time and place in gardens, but we do need to find efficient and effective ways to make sure that they don't cross with wild populations, and also to evaluate them for their value to wildlife, because oftentimes these plants are sold as natives and, you know, beneficial to pollinators, but until all of these studies are done, we can't really say that all of these plants have been, you know, evaluated one by one for their value to pollinators. And we also wanted to end with some acknowledgements. So our lab and our research is supported by Sherry Shang and Spike Wadsworth, as well as grants from the ODA, the Garden Club of America, and we've even had a grant from the NPSO. This slide also has a list of nurseries where we purchased plants for our studies and photo credits. And yeah, so I see there are some questions in the chat, so we can move on to that. And I think somebody else was going to moderate this part.
Yep, I have
Okay.
that, Jen. If you want to maybe stop sharing your screen.
Okay.
Just,
Yeah, and I can pull stuff up if people have
And-
questions based on a specific slide as well.
Yeah, I'll invite everybody to turn on their screens if they're video, if they'd like to. Well, first I want to say thank you, Gail and Jen, for a great presentation. We did have a couple questions in the chat from Marcy and Amy, and I think they were addressed. Maybe I could read them just in case everybody didn't see them. And maybe Gail, you can respond again, perhaps. Or there was, the first question was from Amy. Are there any of the top 10 plants deer resistant? And
I'm sorry. So when I'm like just sitting and listening, I can't help it. I have to answer when a question comes across.
that's okay.
So
Yeah.
really, Jen, I think you can address whether or not you saw deer damage on your plants. On Aaron's plants, honestly, gophers were a bigger problem than deer at his site.
Yeah, the only issues we had with deer were early spring on the camas leaves that were first emerging, and then also on Sidalcia, but we had to kick Sidalcia out of the study for other reasons. And everything else, I don't think we ever saw any nibbling from.
Great. The second question was from Marcy. Let me get to that. Was in Jen's study, are we talking about all pollinators and not taking into account whether the pollinator is native or not?
So I guess the easy answer is both. So I have like a million subsets of my data in the field, and in our like actual sampling, we collect both native and non-native pollinators. But for my different analyses, I can add and remove those groups. So again, we're really interested in like native plants for native bees. So for a lot of our analyses, we take honeybees out because we don't really care about them. And then there are other non-native bees, such as like Anthidium manicatum, which we're able to ID in the field. And so we can take it out of those calculations as well. So I try to say when I'm talking about specific data, what that data set is based on. So for the two slides that I kind of ended on with 2021 and 2020, that was just native bee observations. And then that little extra that I added is when we add back other pollinators and non-native pollinators.
Thank you. I have a question. So I'm a big fan of your top 10 list. I love Pearlies. I like Gilia, Farewell to Spring, and all those Asteraceae. But I also do like lavender in my garden. Is there a detriment to planting lavender with those natives? Does it prevent? Is there a conflict with native bees and the non-native bees, or does it not really have an effect on that?
So I think the biggest detriment that I see from planting lavender is that it gives many gardeners who aren't as familiar with what types of different bees there are and what native bees look like. It gives many gardeners a false sense that they're doing bee-friendly gardening when really they're just doing honeybee gardening. We actually have like a pet hypothesis that we're tending about lavender, that it might be possible to use lavender as a trap plant, if you will, to draw the honeybees to lavender, to leave like the good stuff, the native plants for the native bees. But that's just an idea at this point.
Oh, you're muted, Stephen.
Thank you, Gail. That's interesting. Does anyone else have any other questions? I do want to mention there is a number. I posted the Oregon Nursery web. We have a site on the Native Plant Society page at the state level where we have a list of nurseries and seed growers, and then some other participants also put their suggestions. We do have another question here from Mark. Do you know what vegetative successional state Douglas aster is in? Is it an early pioneer ground cover like dandelion, or can it tolerate a higher fungal soil like other perennials?
We both unmuted. Do you have a good answer, Gail?
I don't. I was going to say off the top of my head, I don't know. I think based upon what I've seen with NRCS recommendations for using Doug aster in restoration work, that it's not an early successional species, that it might be kind of like stage 2 or stage 3 in ecological succession, but I don't know that for sure.
I can address that a bit. The Douglas aster in our yard for the last 7 years has come up and just flourished. And so it's fabulous this time of year. And we have it in our front yard, which is a deer zone. So the deer have not bothered it. So I highly recommend Douglas aster, the native Douglas aster.
Yeah, the only thing that, okay. The only thing that I can add to that is I will say both the natives and especially the cultivars of the Douglas aster are like crazy robust. Like we had to stop watering them because I kept telling my undergraduates that they were getting too powerful because they were just shooting rhizomes into the plots next door to them. And then I'm honestly kind of afraid to go back after these rains that we've had, because after just a little bit of rain at night in Corvallis, their numbers of flowers were actually starting to drop in the beginning of September, and now they're like well above their original like early September bloom count. So it's like I don't know if they're ever going to be able to get rid of them when we're done with our project. So I'd be really impressed to find an ecosystem where they did not thrive. And then Marcy, I did see your question in the chat about the Fender's blue butterfly. And so my understanding of basically how it's become a conservation concern is not only just generally like habitat loss for the butterfly, but Kincaid's lupin will readily interbreed with other lupin species. And this is not just like cultivar specifically, other native lupins, I guess it can cross with, and the butterfly itself will not use the hosts or use the hybrids as hosts. And so because it readily hybridizes, that even limits the genotype of the original plant that is available to create new true offspring from the plants.
Ooh.
So thank you. Marcy, I think that answered your question. You had your hand up, so I think it was for that question. Anyone
Yes.
else have any other questions for our speakers tonight? Well, can I get everybody to give a round of applause to Gail and Jen tonight for a wonderful program. Thank you
Thank you
so
so
much.
much.
For your time.
Yay.
Like I mentioned earlier, this program was recorded, so if you'd like to watch it again or share it with a friend, please visit our YouTube site. And yeah, thank you so much, Gail and Jen. Really appreciate your time today. Excellent presentation.
Thank you for having us.
It was great. Thank you so much.
Thanks, folks.
Is it over?
Thanks, everybody. See you next time.
Thanks, everybody, for joining us in this wonderful presentation.
Thanks, NPSO. You're great.